This method will help elucidate molecular mechanisms of multifaceted biological phenomena, such as mechanobiology, optical electrophysiology, and super-resolution DNA, RNA, protein imaging, using automatic live cell imaging techniques.
This protocol enables automatic, multifunctional, high-throughput, self-drift correcting, and long-term image acquisition. It is compatible with most fluorescent microscopy platforms, such as Nikon. Anyone who tries this technique for the first time should understand the function of each step instead of only following the protocol strictly to ensure a successful experiment.
Begin by placing the environment chamber onto the motorized stage of the inverted microscope. Set the CO2 flow rate to 160 milliliters per minute and adjust the temperature of the chamber. Then add 40 milliliters of purified water into the bath of the chamber. Take out the glass-bottom petri dish with cells from the incubator and place it in the environment chamber. Turn on the confocal controller in the inverted microscope. Switch the light path to the right. Observe the cells attaching using Micromanager. If sufficient cells have been attached to the gel, transfer the petri dish back to the incubator. If not, continue the cell incubation for another 30 minutes for B2B and 60 minutes for lung cancer or PC9 cells. Cut 2 small pieces of adhesive tape and stick them on the chamber around the circular hole. Then, apply a little adhesive glue onto the area of the tape that the petri dish will cover. Take out the petri dish from the incubator.